Hi, my name is Ron Najafi. I'm the CEO of Emery Pharma. It's an honor to have Professor Olson here today, virtually. Professor David E. Olson studied chemistry and neurosciences at Stanford University and the Stanley Center for Psychiatric Research at the Broad Institute of MIT and Harvard. His academic lab at UC Davis discovered that psychedelic promotes structural and functional neuroplasticity in the cortex. They coined the term psychoplastogen to describe small molecules that produce rapid and long-lasting psychedelic and ketamine-like effect on neuronal structure after a single dose. And they invented the non-hallucinogenic psychoplastogen capable of producing sustained therapeutic effect in preclinical model after a single dose. Professor Olson's expertise spanned central nervous system, medicinal chemistry, molecular, cellular neurobiology, and behavioral neuropharmacology. He's an associate editor at the ACS Chemical Neuroscience and has received numerous awards, including the Geordie Folch Pye Award from the American Society of Neurochemistry, the Sigma Chi Young Investigator Award, the Life Young Investigator Award, Sacramento Business Journal's 40 Under 40, among many others. He is the founding director of the UC Davis Institute for Psychedelic and Neurotherapeutics and is the co-founder and the chief innovation officer of Decoupling Neurotherapeutics. In 2019, myself personally, and my family, we lost our father to Alzheimer and dementia. It was then that we realized how little medical intervention we had at our disposal. It was like watching my father slip away out of our hands. That's when my family established the Najafi Award at UC Davis Department of Chemistry. The faculty and postdoc at UC Davis have a chance to compete for a $30,000 annual grant. The research application needs to be focused on sparking innovation at the interface of chemistry, biology, and neurology with the hope of developing drugs for CNS diseases. Our first year recipient for the award, which was back in 2020, was Professor David Olson. So it is our pleasure to host David today. I had a chance to review his slides, and I can assure you there are lots of gold nuggets within his presentation, so stay tuned. Here is Dr. Olson.
Thanks, Ron. And before I start, I just want to thank you and your family again for your generous support of our research. I'm very hopeful that some of the discoveries that we've made will eventually make it into human trials and hopefully impact the lives of patients. So before we begin today, I need to tell you that in addition to my academic appointments at UC Davis, I'm also a co-founder and the chief innovation officer of Neurotherapeutics, but all the work I'm going to tell you about today is work done in my academic laboratory. So our group is really interested in finding new treatments for a variety of brain disorders, but today I'm going to focus on the so-called stress-related neuropsychiatric diseases. These are conditions like depression, post-traumatic stress disorder, and substance use disorder, conditions that are precipitated or exacerbated by chronic stress. Now, a hallmark of all of these illnesses is physical changes in the structure of the brain, particularly the atrophy of key neurons in the prefrontal cortex. Now, that's perhaps unsurprising because the prefrontal cortex projects to a whole bunch of subcortical regions that regulate things like mood, motivation, fear, and reward. Now, our current therapeutic strategies for treating these diseases, unfortunately, are far from perfect. They have largely relied on trying to rectify chemical imbalances in the brain through the use of things like selective serotonin reuptake inhibitors. But unfortunately, these medicines take weeks to months to demonstrate any kind of efficacy, and about a third of all patients will never respond to these traditional types of medicines. Our group has been trying to pioneer a new approach one that involves the use of small molecules to selectively modify specific circuits in the brain to produce long-lasting behavioral changes. And we reasoned that perhaps one of the best ways to impact the neural circuit was to physically change the structure of neurons within that circuit. And so for the purposes of today's talk, I'm going to be focusing on the structure of these layer 5 pyramidal neurons in the cortex that tend to atrophy in response to chronic stress. So we started studying this class of compounds that we call psychoplastogens. These are just chemical stimuli that induce structural neuroplasticity, such as the growth of these new dendrites. And if you zoom in on one of these dendrites, you'll see the growth of these little tiny nubs called dendritic spines. And dendritic spines are the sites of excitatory synapses in the brain. So these changes in structural plasticity often manifest as functional changes that you can measure with traditional techniques like electrophysiology. Now, one of the best known psychoplastogens is this molecule on the left here, ketamine. Ketamine is a dissociative anesthetic that's been used in medicine for many, many years, but more recently it was approved by the FDA for treatment-resistant depression. And the way that we believe ketamine is working is by promoting the growth of those really critical cortical neurons. So here I'm just showing you some cultured neurons that were treated with ketamine, and you can see they have more of these dendritic branches. And then on the bottom here, these green dots are simply representing synapses. So you can see that ketamine promotes both neuritogenesis as well as synaptogenesis. Now, I'm showing you some work from the late Ron Duman's lab and George Akajanian. And what they were able to demonstrate is that chronic stress leads to that cortical atrophy, the loss of those dendritic spines that I was telling you about, which manifests as synaptic deficits. But what is really remarkable is that a single dose of ketamine can rapidly regrow those lost dendritic spines and reestablish synaptic connectivity in the cortex. So I think that ketamine is super exciting for neuropsychiatry because it's pushing us in a completely new direction, a direction that involves the physical healing of the brain and fixing these damaged neural circuits rather than simply trying to treat disease symptoms. However, ketamine is not a perfect drug. It lacks what I would call circuit level selectivity. One of its primary targets is the NMDA receptor, which is found broadly across the entire brain. And so it has some non-selective effects that are less than ideal. So when we got into this space and we were trying to identify molecules that had ketamine-like effects on neuronal structure, we were trying to think of compounds that would hit targets that are selectively expressed exactly in the regions of the brain that we care the most about. And that led us to study the serotonin or 5-HT2A receptor. The 5-HT2A receptor is heavily expressed on these layer 5 pyramidal neurons in the cortex that tend to atrophy in response to chronic stress. And you can see in the rodent brain, it has high expression in the anterior parts of the brain, with less expression as you move towards the back of the brain and very little expression in structures like the hippocampus or the striatum. Now, if you're interested in trying to modulate the 5-HT2A receptor, one of the first classes of drugs that you think about are the classic serotonergic psychedelics, molecules like LSD and psilocybin, which comes from magic mushrooms. So one of the very first experiments that we did is we simply cultured cortical neurons, treated them with psychedelics, and looked at their morphology. And as you can see here, compared to the vehicle-treated neurons in the middle, the neurons treated with the psychedelics DMT and LSD have substantially more complex dendritic arbors. And if you do some super resolution microscopy and zoom in on one of those dendrites, you'll also see the growth of these dendritic spines and these filopodia-like structures. Now, there's a number of ways that we can quantify structural plasticity in the brain, but I want to focus on one of them because you're going to see a whole bunch of these graphs throughout the talk. We do something called Scholl analysis. It's a pretty simple technique. All we do is we draw these concentric circles of increasing radii starting at the soma of the cell, and we simply count the number of times one of these branches crosses one of these circles. And so you can produce these really beautiful shoal plots like the one on the right, where on the x-axis is the radii of those circles, and on the y-axis is the number of crossings. But typically the way we like to represent the data is simply looking at the maximum number of crossings. So for something like the vehicle, that would be about 7. And for something like DMT, that would be about 11. So looking at these Nmax plots, we can see that psychedelics from 3 distinct chemical classes, ergolines like LSD, tryptamines like DMT, and amphetamines like DOI, they're all really good at increasing the complexity of dendritic arbors. Now, for all of these experiments that we were doing at this point, we were treating the cells for 3 days and then looking at structure. And we know that in vivo, that's not what's going on. In vivo, you give a psychedelic, it rapidly gets into the brain, and then it's rapidly cleared. But they seem to produce these very long-lasting effects. And so we were wondering, what is the shortest stimulation period that we could get away with and still produce long-lasting changes in neuronal structure? So instead of stimulating for 3 days, we just stimulated for 24 hours or an hour or just 15 minutes. And then we take the drug away and give the neurons time to grow. And when we do that, we find that even this 15-minute stimulation is sufficient to produce a long-lasting increase in dendritic growth. Now, you have to give the neurons time to grow. If you stimulate for an hour, fix and stain the cells in the image, you see no changes in cortical neuron structure. But if you give them time to grow, you'll see very robust changes. Now, in order to demonstrate this in vivo, we relied on a very rapidly cleared psychedelic, dimethyltryptamine. The half-life of DMT is only about 10 minutes in a rodent. And so for this experiment, what we did is we gave a single IP injection of DMT, waited 24 hours, a time period when all of the drug has been cleared from the body. We then sacrifice the animal, take tissue, and look at structure via Golgi staining and function via electrophysiology. And we find that even after the drug has been cleared from the body, we see this very robust increase in dendritic spine density. And we also see changes in functional plasticity. In particular, we see an increase in both the amplitude and the frequency of these spontaneous excitatory postsynaptic currents. Now, ever since we published this work way back in 2018, it has been replicated and extended by a number of groups across the world. But I want to focus on one study in particular because I think it's quite remarkable. And this was a study done by Alex Kwan's group when he was at Yale. He has since moved to Cornell. And Alex was really interested to see just how long do these effects last for. And he gave a single dose of psilocybin to male and female mice. And he found that even after a single dose, he saw increases in dendritic spine density that lasted for at least a month, which is quite remarkable. Now, in addition to their long-lasting sustained effects after a single dose, the other thing that's really unique about psychedelics is that they seem to have this transdiagnostic panacea-like property. They work across a variety of indications: depression, PTSD, substance use disorder. And I want to emphasize that these drugs are not panaceas. We think the reason they work across indications is because they're targeting a very critical hub in the brain, the prefrontal cortex. And we know from a whole bunch of opto and chemogenetic studies that there are circuits that originate in the prefrontal cortex that do things like regulate drug-seeking behavior, control the expression of fear memories, impact motivation, etc. And so to summarize a lot of work very, very quickly, in general, what we find is that psychoplastogens produce long-lasting behavioral effects in behaviors that are known to involve PFC circuitry, such as the enhancement of fear extinction learning, or producing antidepressant-like effects in something like the forced swim test. Now, at this point, we were very interested in trying to understand the mechanism by which these drugs produce their effects on cortical neuron structure. And so we started with some selective antagonist studies. And we found that if we used an antagonist of the 5-HT2A receptor, we were able to block the effects. The 5-HT2A receptor is known to mediate the hallucinogenic effects of psychedelics. And so this is one of the first receptors we looked at. We also wanted to probe a couple of downstream kinases that had been implicated in the mechanism of ketamine. One is TrkB, which is the high affinity receptor for brain-derived neurotrophic factor, and the other is mTOR, which is necessary for the production of all of the proteins that are required for a neuron to change its morphology and to change its synaptic weights, all those structural proteins and those ion channels. Previous work had shown that if you block mTOR or TrkB, you block the effects of ketamine, and we see something very similar here with psychedelics. Now, at this point, we realized that the antagonist that we were using to block the 2A receptor was not perfectly selective. It hits a couple of other receptors. So to be really, really certain that it was the 2A receptor mediating these effects, we relied on genetic knockout models. So here I'm showing you in wild type animals, when you treat them with the psychedelic 5-methoxy-DMT, you see this really nice increase in dendritic spine density in the PFC. But in the knockout animals, that effect is completely gone. You see very similar things in terms of functional plasticity. Again, wild type animals, you get a robust effect in functional plasticity, but in the knockout animals, the effects are gone. So to determine the role of the 2A receptor in behavior, we ran an experiment relevant to depression. And this is a particular type of the forced swim test that is a little more challenging, I would say. Traditional antidepressants tend to not work in this, but these next generation psychoplastogens tend to be more effective. So on the first day, we run a forced swim test to kind of stress the animals out. And you can see that all the groups at this point are exactly the same. We then give a dose of 5-MeO-DMT, and we do a behavioral assay just to make sure that the dose is getting into the brain and doing what we expect. But then 24 hours later, after the drug has been cleared from the body, we assess the antidepressant potential. And you can see that in the absence of the 5-HT2 antagonist, ketanserin, 5-MeO-DMT causes this robust antidepressant response. But if you pre-treat with the antagonist, you can completely block that. Now, in addition to looking at forced swim test behavior, which is relevant to, I would say, motivated behaviors, we also wanted to assess anhedonia. And the way we do this in mice is using a sucrose preference test. So mice, much like myself, will prefer a sugary beverage over just plain old water. And you can measure this preference. And you can see that in wild type and the 5-HT2A knockout animals, you have a really robust preference for the sugary sucrose containing solution. But you can induce an anhedonia-like phenotype by giving chronic stress. And in this case, we're giving chronic corticosterone injections. And then we can do another sucrose preference test on day 12. And you can see this big reduction in their sucrose preference. Now, what is really interesting is that on day 13, we give a single dose of psilocybin, come back on day 14 and look for sucrose preference. And we can see that in the wild type animals, we see a complete rescue of the sucrose preference, but that effect is completely gone in the 5-HT2A knockout animals. So at this point, we're feeling pretty confident that the 5-HT2A receptor is mediating the psychoplastogenic effects of the compounds and at least some of their antidepressant behavioral responses. But there was a piece of data that really bothered us. And that was the fact that this molecule here on the left, serotonin, could not promote cortical neuron growth. And that had us scratching our heads for a long time because serotonin is a very good agonist of the serotonin 2A receptor. And so this didn't really make a whole lot of sense until we did this structure-activity relationship study. Basically, we were making small structural changes to serotonin-like molecules, and we found that as we started to add methyl groups, going from one methyl group to two methyl groups, we saw an increase in the ability of these molecules to promote cortical neuron growth. And if you look at the Scholl plots, it's pretty pretty striking. Going from tryptamine, you see no difference compared to vehicle. N-methyltryptamine, you see a little bit, but DMT, dimethyltryptamine, you see a very, very big bump. So then we tried to correlate activation of the 5-HT2A receptor with efficacy in this neuroplasticity assay, and we didn't see any efficacy. If anything, we saw maybe a negative correlation. But then we correlated the lipophilicity of the drugs, and we saw a really nice positive correlation with their ability to promote cortical neuron growth. And this led us to the hypothesis that maybe the reason that serotonin cannot promote cortical neuron growth is because maybe it can't access the target. Maybe this particular G protein coupled receptor is not existing on the cell surface. Maybe it's existing on the inside of the cells, and serotonin is simply too polar to cross the nonpolar plasma membrane and access that receptor population. But the psychedelics, which are very lipophilic, can easily cross the membranes. So to start to address this question, the very first thing we did is simply look at where the receptor is localized. So for this experiment, we simply expressed the 5-HT2A receptor and the beta-2 adrenergic receptor in hex cells. They're tagged with CFP, so we use CFP as a control. You can see that CFP is expressed everywhere in the cells, but the GPCRs are expressed pretty much where you would expect them to be. There's a little bit intracellular, but there's a lot kind of on the outside of the cells. And here's just a membrane marker that indicates the boundaries of the cells. But when you express these two GPCRs in cortical neurons, you see something very different. You see that for the 5-HT2A receptors, you primarily see an intracellular localization of the receptors, whereas the beta-2 adrenergic receptor is still expressed on the cell membrane quite a lot. And so we did a whole bunch of follow-up studies to kind of see what cellular organelles that the 5-HT2A receptor might be colocalizing with. And what we found is that when we used this marker of the Golgi complex, we saw a really nice overlap with 5-HT2A receptors in these cortical neurons. But when you look in hex cells, you don't see as good of an overlap. You can see here, there's lots of 5-HT2A receptors that don't overlap with the Golgi, for instance. So we think that in neurons, cortical neurons, the 5-HT2A receptor is existing primarily inside the cell and primarily at the Golgi. Now, I should emphasize, we're not the first to discover this localization. This has been known for many, many, many years. But no one really appreciated what the functional consequence of that intracellular population was. So to probe that, we first developed a few chemical tools. So we started by just taking these membrane permeable compounds in green, two agonists here and an antagonist here, and we just converted them into membrane impermeable versions that are permanently charged. So we just methylated DMT to make trimethyltryptamine, put a phosphate group on this hydroxyl to make psilocybin, and methylated this nitrogen to make methylated ketanserin. And then we simply run the assays under two sets of conditions. The first set is just normal neurons. The second set is under electroporation conditions. And what we do there is we transiently apply a voltage. And what that does is it opens pores in the membrane. That allows the impermeable compounds to cross the membrane. The pores then close, and then the impermeable compounds get stuck on the inside of the cells. When we run the assay without electroporation, we get exactly what you would expect. The permeable compounds, agonists, DMT and psilocin, can promote cortical neuron growth. The impermeable compounds, TMT and psilocybin, cannot. Similarly, the membrane permeable antagonists can block DMT-induced plasticity. The impermeable antagonists cannot. But when we electroporate the cells, we see something very, very different. In this case, we can see the impermeable agonists, TMT and psilocybin, they can promote neuronal growth, and the impermeable antagonist can block cortical neuron growth. So that really suggested that the target is on the inside of the cell. Now, this is a neuritogenesis assay. We did the same thing for a spinogenesis assay. And again, We see the permeable compounds in green promote growth. The impermeable compounds do not. Now, at this point, we reasoned that if it's all about the location of the receptor, serotonin should be able to promote growth if we could just get it to the target. And so for this set of experiments, we relied on sparsely transfecting cortical neurons with a transporter of serotonin, this protein called SERT. So I love this experimental design because within the same culture, you can compare the two different genotypes. So I'm showing you here two neurons side by side. The one on the left here expresses the transporter. The other one doesn't. Similarly, these two neurons here, the one on the right expresses the transporter and the one on the left doesn't. And so what we see is that when you treat neurons of either genotype, again, in the same culture with vehicle, you see no change. When you treat them with DMT, you see increased growth in both genotypes, which makes sense because DMT can just passively cross the cell membrane. But when you treat them with serotonin, you only see growth in the neurons capable of importing serotonin. Now, to follow up on this experiment in a little more detail, we ran this study in the presence of a 5-HT2A antagonist and a blocker of the serotonin transporter. So what I'm showing you here in this bar graph, this is essentially a replication of the last slide that I just showed you. Serotonin can promote growth in SERT-positive neurons only. But then if you treat with ketanserin, you can block this effect, suggesting it's 5-HT2A mediated. And you can also block the effect with citalopram, suggesting that serotonin needs to enter through the transporter in order to turn on the pathway. But when you do this with DMT instead of serotonin, now you see again a replication of that last study. It works in both genotypes. You see that ketanserin blocks the effects, again, because it requires 2A. But citalopram no longer can block the effects of DMT because DMT does not require the transporter to access the 2A receptor. Now, those were neuritogenesis experiments. We did the same thing with spinogenesis experiments. And so again, here I'm showing you that DMT promotes spinogenesis in both genotypes, but serotonin can only promote spine growth in cells capable of importing serotonin. Now, to demonstrate this in vivo, we relied on using a virus to express the serotonin transporter on those cortical neurons. And so normally they don't have the transporter, so they can't import serotonin. But after 3 weeks to allow for viral expression, we then give a single dose of this compound, parachloroamphetamine. It's a serotonin releasing agent. It causes presynaptic release of serotonin. And then we simply do spine analysis 24 hours later. And what we find is that in the neurons that have the transporter, we see a really nice increase in spinogenesis compared to the neurons that lack the transporter. Now, if the spine growth is responsible for the antidepressant effects, we would expect to see something similar. So we ran a very similar experiment, except for after expression of the transporter, we ran a novelty-induced locomotion assay just to show there's no difference between the genotypes, and there wasn't. We then run a forced swim test, again, to show no difference between the genotypes. We then give parachloroamphetamine, come back 24 hours later and measure antidepressant-like effects. And what we see is we get an antidepressant response only in the animals that have the ability to import serotonin into those cortical neurons. Okay. So at this point, we're feeling pretty confident that the intracellular population of 5-HT2A receptors is really critical for psychedelic-induced neuroplasticity and the antidepressant effects. However, there's a problem. And the problem is that the 5-HT2A receptor also mediates the hallucinogenic effects of psychedelics. And while I definitely think these first-generation hallucinogenic medicines are going to help some people, their clinical scalability is limited. These compounds, in order to be administered safely, have to be given under the supervision of a healthcare provider, which drastically increases the cost and the complexity of the treatments. And so we were wondering if it might be possible to decouple the hallucinogenic effects of the drugs from their effects on neuroplasticity and their beneficial effects on behavior. And we thought it might be possible because there were known agonists of the 5-HT2A receptor that are non-hallucinogenic. This receptor exhibits a high degree of functional selectivity. And so in order to try to identify these non-hallucinogenic psychoplastogens, we needed an assay that was very good at predicting hallucinogenic potential. And so we turned to this mouse assay called the head twitch response. You can see the behavior there on the left. It's this rapid rotational movement of the head. And it's very characteristic of serotonergic psychedelics. And so what Adam Halberstadt's group was able to show is that across over 30 structurally distinct psychedelic compounds, there's a really beautiful correlation between human hallucinogenic potency and potency in this head twitch response assay. And so I'll give you an example of kind of just how small the structural change can be in order to completely change the hallucinogenic properties of a molecule. So we started with this compound DMT, a very potent hallucinogen, and we converted it into this iso-DMT analog. You can see here in terms of its psychoplastogenicity compared to ketamine as the positive control, the compounds are essentially the same. They have, equipotent. But their hallucinogenic potentials are very different. So I'm showing you a couple examples here. 5-MeO DMT in blue and 6-MeO iso-DMT in orange. If you had a space-filling model of these two compounds and you're holding them in your hand, you would have a really hard time telling them apart. The only difference really is that this 5-MeO DMT has the ability to engage in a hydrogen bond donor event, which is gone in the iso-DMT counterpart. And even though they have comparable effects on neuroplasticity, they're very different effects in the head twitch assay. 5-MeO DMT causes this nice dose-dependent increase in head twitch response. You don't see that with the 6-MeO iso-DMT analog. Now, the head twitch response is great because it's highly predictive, but it's not very useful for drug discovery, mainly because the throughput is quite low. And so we realized that we needed to develop a cellular assay for hallucinogenic potential. And the way we did this was actually quite simple. We took the serotonin 2A receptor, which again is known to mediate the hallucinogenic effects of psychedelics, and we simply lopped off the third intracellular loop of this GPCR, and we fused on a circular permuted green fluorescent protein. Now, the way these CpGFPs work is there's basically a cut in the beta barrel that allows it to be open. That enables water to get in, quench the fluorescence of the fluorophore. But when a ligand binds to the receptor, it can induce a conformational change that closes the beta barrel, protects the fluorophore, and allows fluorescence to turn on. So you can see that this agonist turns on the sensor really, really nicely, but these two antagonist compounds either do nothing or they actually turn it off slightly. And so we got very excited when we found that potency in this biosensor assay correlated really nicely with human hallucinogenic potency. So we started calling the biosensor Psychlite. Now, one of the first experiments that we did with Psychlite is we used a virus to express it in the prefrontal cortex. We then gave a single administration of 5-methoxy-DMT. You can see that very rapidly Psychlite fluorescence turns on. But what's interesting is it turns on at the same time that the head twitch behavioral response turns on. But what was kind of surprising to us is that the head twitch behavioral response wanes over time, whereas Psychlite fluorescence remains elevated. Now, if you give an injection of the vehicle or you give an injection of the antagonist, you see no change and a decrease in fluorescence respectively. Now, I would say that probably the most exciting day came when we saw these data and we were comparing hallucinogenic compounds in green with their non-hallucinogenic congeners in purple. And you can see that the difference between LSD and glyceride is subtle. Glyceride simply has a nitrogen inserted in here and that stereo center kind of switched, and that's it. In 5-MeO and 6-MeO DMT, the only difference is you move the methoxy group one position over on the indole ring, and that's it. But what is remarkable is that the hallucinogenic compounds turn on the biosensor, the non-hallucinogenic compounds do not. Now, You might be asking yourself, maybe the non-hallucinogenic compounds are not binding to the sensor. But to address that question, we can simply run the assay in antagonist mode. We can give a little bit of agonists to turn on the biosensor, and then titrate in increasing quantities of the non-hallucinogenic ligand, and you can watch the fluorescence intensity decrease. And that's what you see here on the right. So to facilitate our drug discovery efforts, we simply made a stable cell line that expresses cyclite. We call these Cili2 cells. These Cili2 cells are really nice because they exhibit some baseline fluorescence. So inverse agonists turn off the fluorescence and agonists turn on the fluorescence. And we tested a whole bunch of compounds in this assay. I'm showing you on the x-axis is agonist mode, on the y-axis is antagonist mode, and this black dot right here, that's the vehicle control. So compounds that increase cyclite fluorescence more than 1 standard deviation from the vehicle, these are known hallucinogenic compounds in red. Compounds that decrease cyclite fluorescence more than 1 standard deviation in antagonist mode, these are known non-hallucinogenic compounds in blue. And we can simply mathematically combine agonist and antagonist mode data to produce this ligand score on the left here. You can see that compounds with a red ligand score are predicted to be hallucinogenic. Compounds with a black ligand score are predicted to just not bind to the sensor, period. And compounds in blue are predicted to be non-hallucinogenic ligands. What's really interesting about this class is you'll see traditional antagonists like the MDL compound, which is also known as volanerancerin, ketanserin, and antipsychotics like haloperidol. But you'll also see these non-hallucinogenic agonists like glyceride. Now I want to spend just a little bit of time telling you about these two compounds. I'll start with 5-bromo-DMT. What was really interesting about 5-bromo-DMT was that we had synthesized the entire halogenated series going from fluoro to chloro to bromo, just increasing the van der Waals radii of those atoms. And the biosensor predicted the fluoro and the chloro compound to be hallucinogenic and predicted the bromo compound to be non-hallucinogenic. And that's exactly what we see in the behavioral assays. The fluoro compound produces robust head twitches at all doses. The chloro compound produced this nice dose-dependent increase, but the bromo compound did not produce head twitches at any dose tested. Now, we also found this compound that we call AAZ, named after the student who synthesized it. Kind of looks a little like 5-MeO-DMT. The biosensor was predicting that it was going to be non-hallucinogenic, and it didn't produce a head twitch up to even these massive 100 mg per kg doses. Now, a therapeutic dose of AAZ is a lot closer to 10 mg per kg. Now, when we tested this in assays relevant to depression head-to-head with ketamine, we found something really interesting. We found that AAZ had comparable potency to ketamine in vitro for inducing cortical neuron growth. We also found that it had rapid after 30 minutes and sustained lasting at least a week in the forced swim test that was comparable to ketamine. And then finally, we used this genetic model of depression developed by Bill Wetzel's group at Duke, these VMAT2 heterozygous mice. These mice just have some baseline depressive phenotypes. And here I'm showing you that sucrose preference test again. And you can see that the wild type mice have a very robust sucrose preference, but the heterozygous mice do not. A single dose of AAZ can rapidly rescue this anhedonia. And what's really remarkable is that the effect lasts for quite a long time. It lasts for at least a couple of weeks before the genotypes start to differentiate from each other. Okay. So at this point, we have been playing these kind of medicinal chemistry tricks on compounds from every single major psychedelic class. Tryptamines like psilocybin, amphetamines like, DOI, MDMA, ergolines like LSD. But I want to spend a little bit of time to tell you about our work on this compound right here, Ibogaine. Now, Ibogaine is not a traditional classic psychedelic. You might call it an atypical psychedelic. But Ibogaine has some pretty remarkable properties, at least as anecdotal reports and some open label clinical trials would suggest. Those studies have suggested that a single dose of Ibogaine can have anti-addictive properties that last for at least 6 months. And if you take a second dose, they can last up to 3 years. And that is just really remarkable and really indicates plasticity mechanisms to me. And so you might be asking yourself, well, if Ibogaine is so efficacious, why are we not using it to treat the opioid epidemic in this country? And the answer is Ibogaine is a terrible drug for a variety of reasons. Number one, it's hard to come by. There are no de novo total syntheses that can produce Ibogaine enantioselectively in the quantities required for clinical trials, and getting it from the natural sources has a whole bunch of problems associated with that. Number two, Ibogaine is incredibly greasy and it's very insoluble in water, which makes formulation quite challenging. Of course, it has these hallucinogenic properties, but the worst thing about Ibogaine is that it binds to hERG channels in the heart and causes cardiac arrhythmias. And several people have actually died during Ibogaine treatment for their opioid use disorders. So our approach to this problem was to use something called function-oriented synthesis. And this is a really simple concept popularized by Paul Wender at Stanford. And the idea is, can you take a complex structure like Ibogaine and distill it down into the features that are essential for the properties that you care about? And if you look at the structure of Ibogaine, it really has kind of 3 distinct structural motifs. It's got this indole group, it's got this 7-membered tetrahydroazepine ring, and it's got this bicyclic isoquinoline. So all we did was to simply start deleting bonds. So if you delete the bond between the indole and the isoquinoline, you arrive at compounds that look like scaffold 1. If you delete the isoquinoline altogether, you arrive at compounds that look like scaffold 2. And I'm not going to take you through all of the medicinal chemistry that we did. I'm just going to kind of show you some of the high level results. And basically what we found was that compounds that retained the azepine and lacked the isoquinoline, well, they were pretty good for the most part at promoting cortical neuron growth. But the opposite wasn't true. Compounds that contained the isoquinoline but lacked the azepine, they were pretty bad for the most part. So we decided to prioritize this compound here for further study. We made one more small structural tweak to it. We moved the methoxy group 1 position over to produce this compound. The compound on the left looks just like Ibogaine, except it lacks the isoquinoline, so we started calling it Ibogainalog or IBG. But the compound on the right looks a lot like the natural product tabernanthine, so we started calling this tabernanthalog or TBG. Now, unlike Ibogaine, TBG can be synthesized in just a single step in gram quantities using this Fischer-Indole cyclization, and it has a lot of really desirable properties. First and foremost, it's completely soluble in water, which makes handling it really easy. It is still a potent agonist of the 5-HT2A receptor, and it has some other unique properties. And so here is one example that I'm going to show. I want to just emphasize this particular structure-activity relationship study because I think it's really quite remarkable. So 5-methoxy dimethyltryptamine is a substructure within Ibogaine, and it's a very potent hallucinogenic compound. And you can see that 5-MeO-DMT produces a very robust head twitch response. But if you simply conformationally restrict the dimethylamino group to the 2 position of the indole to make IBG, you can see that you drastically lower the hallucinogenic potential. And if you move the methoxy group 1 position over to make TBG, now you've completely eliminated the hallucinogenic potential. So what about the cardiotoxicity? So the other thing that was really interesting about TBG is that by cutting away all that extra grease in the isoquinoline, we drastically reduced the potency of TBG for hERG. So Ibogaine's potency at hERG is about 1 micromolar. You can see that TBG is 150-fold less potent. And you can actually visualize this using these immobilized zebrafish here on the right. So you can see the heartbeat of a vehicle-treated fish. You can see the slowing or the bradycardia induced by Ibogaine, but you just don't see that with TBG-treated fish. Now, one way to measure the propensity of a drug to induce a cardiac arrhythmia is simply to take a ratio of the atrium to ventricle beats per minute. And here we see that Ibogaine is very good at causing cardiac arrhythmias, as is the positive control serindole, but TBG looks just like the vehicle control. Now, in addition to the cardiotoxicity, TBG has some just general toxicity issues associated with it. And you can look at these pie charts, but I think a picture is worth 1,000 words. This is what a vehicle-treated fish looks like. This is what an Ibogaine-treated fish looks like. They look very different. But a TBG-treated fish looks just like the vehicle control. Now, it's great that we produced a safer version of Ibogaine, but none of this matters if it doesn't have Ibogaine-like efficacy. And so the first thing we did is verify in vitro that TBG is a potent psychoplastogen, but this is an in vivo study looking at that same effect. And the way this works is we have these mice that are genetically modified to have their neurons in the cortex labeled with a fluorescent protein so we can visualize them. We simply cut a window in the skull and we use a 2-photon microscope to look at a section of dendrite. We then give a single dose of TBG, come back 24 hours later and look at the exact same section of dendrite. And that allows us to quantify the number of spines that were formed, like this one in blue, and the number of spines that were eliminated, like this one in red. And we find that TBG does not really impact spine elimination, but it does seem to increase spine formation, and it does so to a comparable extent as the hallucinogenic drug DOI. Now, again, if the spinogenic effects of the compound are really important for the therapeutic effects, we would expect to see kind of broad behavioral changes, and that's exactly what we see. We see that TBG has antidepressant-like effects in the forced swim test, comparable to ketamine. We also see a very robust anti-addictive response in this 2-bottle choice paradigm relevant to treating alcohol use disorder. And then finally, we used a model of heroin use disorder, and we gave TBG at 3 different time points: during self-administration of heroin, during the early stages of extinction or withdrawal, and then immediately prior to cued reinstatement. And it didn't really matter when we gave the drug. We saw this long-lasting protective effect on heroin-seeking behavior. Now, finally, I just want to emphasize that TBG has the potential to actually heal damaged neural circuits. And so what I'm showing you here is some calcium imaging from the somatosensory cortex of mice. Now, mice are very tactile creatures. They like to explore their environment with their whiskers. And every time they whisk, you see this really nice coordinated calcium response in the somatosensory cortex. However, unpredictable mild stress can completely disrupt this coordinated response. But what is really remarkable is that a single administration of TBG, long after the drug has been cleared from the body, can completely restore this coordinated response, suggesting that it has healed this damaged neural circuit. So to summarize, what I tried to tell you about today is that in the healthy brain, in your cortical neurons, there's lots of dendritic branches and lots of these dendritic spines, but in many brain disorders, including many neuropsychiatric diseases and in many neurodegenerative diseases, you see a physical atrophying of the brain, a loss of these dendrites and a loss of these dendritic spines. But psychoplastogens, be they hallucinogenic compounds like psychedelics or non-hallucinogenic compounds like TBG, are very good at regrowing these atrophied dendritic arbors. So finally, to summarize, I told you today that psychedelics are very good at promoting structural neuroplasticity through the activation of this intracellular population of 5-HT2A receptors. I told you that cyclate is very good at predicting hallucinogenic potential, and so it has applications in drug discovery. And I told you that TBG and AAZ are potentially safer psychoplastogens. And I'd just like to finish by thanking these key collaborators that played important roles in this work. In particular, I'd like to thank Lynn Tien. It was a really great collaboration working with her group on the development of Cyclite. And I'd like to thank these funding agencies on the right. In particular, I'd like to thank the Najafi family for their generous gift. Unfortunately, we have not published the work that we did as part of the Najafi Award yet. But I want to tell you right now, compared to everything else that I told you about today, that work is the most exciting. We have a really phenomenal compound. It's currently in revision. And so I'm hoping that maybe in a few months, maybe I can come back to Emory in person and give a lecture on that work. Really, really exciting stuff. We're hopeful that it's going to have some real impact on neurodegenerative diseases. And finally, I just want to thank all of the really incredible students that I get to work with every day. Very creative, talented, hardworking bunch. I've highlighted the names of just a few people whose research I focused on today. And with that, I just want to thank you for your attention, and I'd be happy to answer any questions.
Fantastic present. Thank you so much, David. Questions?
Yeah, I have a question, David. My name is Mark Nelson, and I work with Ron and Kelly, and I run the chemical division of CP Lab Safety. And I've known Ron for years. My question is are there any molecules that are not hallucinogens or have psychotropic activity that cause spine, these dendritic spine maturations that you know of?
Are you talking about in addition to the novel ones that we've invented, you mean?
Yeah, that are outside the psychotropic world. Because I know a little bit about the area because there's these professors that I met years ago at UC Riverside, and they were studying how minocycline, the tetracycline antibiotic, changed dendritic spine maturation rates. And they were studying Fragile X. Do you know the Ethels? I think they're a husband and wife team.
No, I'm not familiar with those researchers, but I can say that in general, there are lots of compounds that impact plasticity in various ways. So for instance, we've studied a natural product called bryostatin. Others have studied it too. Bryostatin is being used for Alzheimer's disease. It's been in phase 2 clinical trials for a while now. It's not a great drug from, the properties of the molecule. It's massive. It doesn't have great brain penetrance, that kind of stuff. But bryostatin is well known to impact structural and functional neuroplasticity. In particular, it promotes synapse maturation and synapse formation. But we've shown that in the cortex, in addition to promoting synapse maturation, it actually causes spine elimination. And we think that might be a way to kind of increase the signal to noise for memory processes. And maybe that's why it has pro-memory effects. But that might be better for, a memory disease versus the physical atrophying in some of these other conditions. So in addition to, the psychoplastogens that we've developed, there are other known non-hallucinogenic compounds that have therapeutic value. Whether or not they can promote spine growth in humans, I mean, that's something that no one really knows. We don't even know if psychedelics do that because it's hard to measure this in humans. But there was just a recent paper for an LSD analog called Bull 148 that came out that's known to be non-hallucinogenic in people, and it has plasticity promoting effects in preclinical species. And Bull was actually used in the clinic to treat cluster headaches, and it had pretty profound effects there. And there's a lot of companies that are interested in seeing what it does for neuropsychiatry.
Sure. I guess my second question related is, do you know what effects some of these more potent and lipophilic hallucinogens have on mitochondria and the signaling processes and all that? Because they're all intimately related, I think.
They sure are. And that's a question that we're currently trying to address with my collaborator, Uri Manor and Vidita Veja. They are both mitochondria experts. And so we're trying to assess that. Now, it's known, Vidita published some work on mitochondrial biogenesis in response to psychedelics. So these compounds definitely impact mitochondrial function. The big question, I think, is that a direct effect or an indirect effect? Is it the plasticity growth that leads to changes in mitochondria, or is it changes in mitochondria that lead to the plasticity? And that's what we're trying to figure out.
Yeah, well, very good. Thank you for your talk.
David, I have 2 questions, kind of unrelated. what is the excitement around psilocybin and microdosing psilocybin? And also, are there models, mouse models or other animal models for completely severed sort of neurons or nerves where you get a complete cut, and are you able to show that you could reconnect or reattach nerves?
So I'll answer the second question first. There are some spinal cord injury models that you can use, and we are interested in doing those studies, and we've been trying to identify the right collaborators to run those. We have not done them yet, so we don't know. But I think it's possible that you might get some restoration or regrowth of these injured nerves. Now, for your microdosing question, actually, I'll show you some data. Oh, I don't have it. It's too bad. So often in presentations, we actually did a microdosing study in rodents, and we gave sub-hallucinogenic doses of DMT every third day to rodents for a period of about 2 weeks. And then we did behavioral tests on the intervening days. And what we found was that for the most part, there were no changes, but we did see an antidepressant effect and an anxiolytic effect in fear extinction learning. Now, I think a lot of people are excited about the possibility of microdosing, but I would say in humans, there's very limited evidence for efficacy. And I think the reason is you got to get the dosing just right. If you're not engaging a certain number of those 5-HT2A receptors in the brain, you're not going to get the plasticity effects. So it seems that there is kind of a threshold for engagement. So if you look at target engagement studies, you'll see that as you go up to about 30 or 40% receptor engagement, you can start to turn on plasticity. But then when you get up to about 60%, you start to turn on hallucinations. And so it's a very, very fine line to do that with a hallucinogenic drug. So some clinical studies have shown modest effects of microdosing. Many of them have shown no effects. And so it could be that psychedelic microdosing is just, a form of a placebo effect. The other thing I want to emphasize, though, is that a lot of people want to do microdosing because they think of it as a safer version than taking a high hallucinogenic dose because it's sub-hallucinogenic. But that's a problem because many of these first generation psychedelic compounds also agonize 5-HT2B receptors in the heart. We all know that is a common off target that leads to cardiac valvulopathy, and several drugs have been pulled from the market because of their 5-HT2B agonism. So the people that are trying to microdose might be doing more harm to themselves than good by causing some long-term issues with their heart function.
Thank you. Any other questions?
Dr. Olson, with your new analogs, I'm assuming when you do the in vivo studies, these are all orally administered? Yes?
I will say that my academic lab focuses on IP administration, but my company obviously checks for oral bioavailability.
I see. Do you have any efflux-related, do you come across any efflux-related issues traversing the blood-brain barrier? Have you come across any of that issues with your new analogs that you're working on?
Yeah, I'll say that. So Delix has licensed a whole bunch of technology from UC Davis and developed their own compounds. They've made, I don't know, over 2,000 different non-hallucinogenic psychoplastogens at this point. And they have a couple of them that are actually going into the clinic. One is actually scheduled to be in the clinic in just a few days. But there are two clinical candidates that they have right now. There's no issue with efflux, and it reaches very, very high brain levels.
Once again, let's thank Dr. Olson. Thank you. Thank you, David, and look forward to seeing you here next time at Emory.
That'd be great.
All right. Thanks again.
Thank you.
Okay. Take care.